So I just want to take one brief step back to consider how we're phrasing things in this scenario. So notice that I'm describing the fish as hypertonic to its environment. And as a result, water wants to enter the fish. Now, another way to think about this, if that statement feels confusing to you in the way that we've discussed this within the first half of the course, is that if the fish is hypertonic to its environment, we have now placed the fish into a hypotonic solution. Okay, so the fish itself is hypertonic to its environment, which means that the surrounding environment is hypotonic to the fish. So this idea of water flow still holds. Okay, we're not redefining any of the things that we've talked about, but I do want you to take a second to think about how we're phrasing things so that there isn't any confusion when we apply the terminology on a lecture exam. This image provides us with a summary of the key concepts for our freshwater fish. So the freshwater fish lives in water less concentrated than the body fluids. And so as a result, the fish has a tendency to gain water, okay, as a result of osmosis, but then lose salt because the concentration inside the fish is higher than the concentration of salt outside. So there would be diffusion down the gradient of salt as well as water. Okay, so diffusion for both down their respective gradients. Now, for a fish like this, because there's already a tendency to gain water, they don't want to take in water purposefully, so they do not drink water. So there will be water that does go in because, again, there is still the tendency for water to enter based on osmosis. And then there will be salt that goes in, and that salt in will be through active transport by the gills. There will be some movement of salt that goes out, some movement of water that goes out. And this idea of movement here is going to result in a large volume of urine, okay? Because if we look at our arrow, we can see that, the width of the arrow is a little bit broader, comparatively speaking, to the other arrows. So we have a large volume of urine, which essentially means a large amount of water is leaving. And this urine is less concentrated than the body fluids. While in most cases, desiccation or severe dehydration would be bad, here would be an example of a particular type of invertebrate that is capable of losing the bulk of its water and still being able to survive, albeit in this dehydrated and essentially dormant state. And these tardigrades are essentially going through the process of anhydrobiosis. And your book goes over how significant of a water loss that it is. You can also see how tiny these guys are, 50 micrometers. And they're sometimes referred to as water bears. So you could even, in essence, go to a puddle of water after it rains and aspirate some of that water, put that on a microscope, and it's entirely possible that you could uncover one in its hydrated state if you have, a microscope that is strong enough to locate it based on its size. Now, when it comes to this, it's really just more of an example that, severe desiccation can be something that certain organisms, more specifically this type of organism, can indeed handle. But in general, when we're thinking about most organisms, they can't handle that, right? So this would be an adaptation to severe losses in water. And the fact that they can do this is really amazing. I think there's still kind of work to be done regarding certain things that this organism has that allows it to exist like this, and it can exist like this for a really long time prior to receiving enough water to go about its normal hydrated state. One thing that is of interest is that even though it can exist for a really long time in a dehydrated state, once it is hydrated, it does have a finite lifespan. So this isn't like an immortal type of organism, but it's just one that shows us a certain type of adaptation based on conditions that it had to kind of respond to. So as I record this particular slide, my dog is literally lapping up water behind me, and it fits, right? Because now we're thinking about land animals or terrestrial vertebrates and how they are able to maintain their balance, right? And when it comes to land animals, they will have a higher concentration of water than the surrounding environment. And in this scenario, the surrounding environment is air. And as a result, there's a tendency to lose that water. And that water loss occurs through the process of evaporation, through the skin, or through the lungs. And different types of land animals will have different approaches in order to accommodate significant water loss, whether that's behaving in a nocturnal fashion, so kind of emerging or utilizing their energy in the nighttime. But ultimately, what this comes down to in terms of water acquisition is to obtain it through food or water. Now, when it comes to the process of osmoregulation, there is still expenditure of energy, and that expenditure of energy is for the purpose of maintaining osmotic gradients. So it's not just all about passively moving from one area to the next. We have to think about the needs of the organism in terms of solute concentration, retention of water. So there is absolutely energy that is involved in these processes. When it comes to the amount of energy that's utilized, it's going to incorporate several factors of which your book outlines. So the amount of energy is going to vary based on the osmolarity that the animal has relative to its surroundings, meaning, like how much work needs to be done in order to maintain the solute concentration within that organism, or just how easily water or solutes can move across the animal surface. Depending on cell types and cell membrane permeability, they can vary. And another factor can be the work that's required to pump. So when we use that term pump, we're talking about active transport, solutes across the membrane. Now, when it comes to our discussion of osmoregulation, we're not going to go through this in quite the same way that I covered this concept in anatomy and physiology. So I'm not going to go through the majority of pumps and channels that are involved in this process. But just know that in order for us to maintain a lot of the things that are going on within this, for moving certain solutes, they're absolutely incorporated. So we'll go through some, but we're really just going to provide just a really broad approach here, and we're not going to go through the same level of detail, but there's lots of proteins that are involved in helping to maintain balance, especially when we think about mammalian osmoregulation. Now, when it comes to osmoregulation, there needs to be certain cells whose job it is to kind of facilitate this process. And we'll think about it with respect to the example of this marine bird, but we'll also see this when we go through mammalian osmoregulation / excretion. So for the marine bird, notice that in the cartoon rendering, it has salt secretions. And this is important because this marine bird is surrounded by salt water, but it still needs to take in water, right? So the idea was, how is this bird able to take in water from the surrounding environment and kind of not deal with the dehydration that one would expect based on that excess amount of solutes that it's ingesting. And so what they discovered was that the birds had a nasal salt gland. And when we kind of zoom in and start to think about its level of organization, you will see parallels between this structure and that of our nephrons, right, or our functional units within the kidneys. And so in this illustration, that we see for the process within the marine bird, what we have here is the capillaries, and we have that's going to allow for blood flow. We have our transport epithelial tissue, and then we have the lumen of whatever the secretory tubule is going to be. So in this example, what we find is that blood is flowing in one direction. And we can see that when we look inside that vessel, there's a ton of solute down here. And there is going to be movement of that solute through the utilization of transport epithelial tissue into the lumen of the secretory tubule. So more salt ions can enter. And as blood continues to flow, we find that the solute concentration begins to decline. Now, when we look at the lumen of the tubule, what we find is that its direction is opposing that of our blood vessel. And it's still receiving solutes, because even if we look at the top, while the solute concentration of the blood vessel has been declining, it's still higher than that of the secretory tubule. So it's still going to promote movement from one side to the next. So we can see as the lumen works its way downwards that the salt concentration rises. And so that now kind of really high solute or highly concentrated fluid is going to be able to flow through that duct and end up coming out of that nasal salt gland that's seen in the marine bird. And so as a result, the marine bird is able to take in that salt water, dispose of a ton of the solutes, and not really have to kind of go into the reservoirs of kind of its excretory system. And so it's kind of depending on these tubular networks that are here in order to remove the excess sodium chloride from the blood. So just in thinking about our take home for this section, this is really all about how osmotic balance is maintained, and the need for osmotic balance is important because tissues depend on that balance to carry out effective metabolic functions. So there were different physiological mechanisms that vertebrates as well as invertebrates will utilize with the goal of keeping their blood osmolarity and their ion concentrations relatively constant. So that's the take home of this section. So there's different examples that are discussed through here, but then you also want to think about how that relates to the mammalian process, which we'll get to in our upcoming modules. Concept 44.2, an animal's nitrogenous waste reflects its phylogeny and habitat. When it comes to this illustration, this is really showing us the downstream products of catabolic reactions. And catabolic reactions are ones in which substances are broken down. Now, the catabolic reaction that is seen in proteins and nucleic acids produces nitrogen-containing byproducts, and those are referred to as nitrogenous waste, and they need to be eliminated from the body. The focus in Bio 100 was on carbohydrate metabolism. So in short, the first step of metabolism of amino acids and nucleic acids is deamination, which is the removal of an amino group. Okay, so we'll go through these different types of nitrogenous waste in separate voice threads. First, we have ammonia. And ammonia we see is a nitrogenous waste seen in a lot of aquatic animals, including most bony fish. When it comes to this nitrogenous waste compared to the others, this is the most toxic. And it's one of the most toxic because ammonia and ammonium ions can disrupt the pH of body fluids, and we want to maintain homeostasis there. It can disrupt electrochemical gradients, and electrochemical gradients are important for communication, facilitating important events within the body, and just a ton of reactions that involve redox, right? So oxidation and reduction. And when it comes to this type of waste, again, it's going to be animals that are going to be primarily aquatic. And the reason behind this is because this needs to be excreted in a really dilute way in order to be safe. It's so toxic that these organisms actually get rid of it almost as quickly as they make it. Now, one may question, okay, well, if this is really toxic, why are they generating it in the first place? But a benefit to that, because we're thinking about how their waste reflect their habitat, is that if they generate this toxic waste, they're actually using very little energy. So that initial process of deamination, you can see here that there's not much change taking place, but we have our waste. So there's less energy that is involved in the production of ammonia, but the disadvantage would be that a ton of body water is needed in order to get rid of it, in order for the ammonia to be diluted enough that it is going to be safe. Next is urea, which is produced from ammonia, and it's produced by all mammals, some amphibians, marine fish, reptiles. And when it comes to urea, it is a little bit better than ammonia because it is going to be a little less toxic. And urea is a water-soluble nitrogenous waste, so it can be excreted within urine, but it doesn't require large volumes of water for its excretion. So making urea helps to conserve some water and removes the need for constant removal and reduces the possibility of toxicity that could occur when ammonia is the nitrogenous waste that's produced. But if we want to think about pros and cons for our nitrogenous waste, then a con to having urea is that it's going to need a little bit more energy for its production. So a moderate expenditure of ATP is required in order for urea to be produced. So there has to be, some budgeting with respect to energy expenditure for this organism towards the production of urea. Our third nitrogenous waste is uric acid, which is only slightly soluble in water. It actually precipitates and can be excreted using very little water. So uric acid forms this really pasty substance, which is what you see when you come across bird droppings. And uric acid is going to be produced by birds as well as insects and most reptiles. In contrast to how some reptiles will produce urea, most will actually depend on uric acid as far as nitrogenous waste products go. Now, when it comes to this type of nitrogenous waste, this has, if we're ranking them, the highest level of energy investment required in order to produce it. So it's going to get balanced against the water conservation because there is not a need for a ton of water here. There's a benefit of conservation of water, but there's also a high level of energy expenditure. So there has to be more that is allocated within the energy budget towards the process of producing this type of waste. Something to observe about the different examples of nitrogenous waste was that they varied with respect to their level of toxicity or the amount of water that was required in order to excrete them. And so what this slide provides is essentially a summary of what was previously discussed, and then there's just a little bit more to factor in as well. So the kind of nitrogenous waste that are released by an animal are going to depend on the evolutionary history and the habitat, but we're going to place our emphasis on availability of water. And keep in mind that ammonia was highly toxic, needed to be very dilute. So we would expect to see that in organisms that have, a large amount of access to water. And when it came to nitrogenous waste, there had to be a factoring in of energy budget. So how much are we allotting with respect to expenditure of energy towards this process? With ammonia being the least amount of energy expenditure, uric acid being the most. Now, something that was not shown within the earlier slide is the immediate environment of the animal egg. And we need to think about why that's important. So one example would be the bird. And when it comes to birds, they have eggs that are encased within a shell, and their nitrogenous waste build up as the embryo grows. Now, the process of uric acid we've discussed generates a precipitate, and that precipitate cannot affect the embryo's development, but it is a significant investment of energy. So here we're seeing the budgeting of energy towards the process far outweighing the idea of another type of nitrogenous base that doesn't form that precipitate that could build up and negatively impact embryonic development. So that would be an example of thinking about the immediate environment of that animal egg. So while this section is short, this really comes down to nitrogenous waste, understanding examples of nitrogenous waste, and based on their form, what that says about how the organism needs to dispose of it. So ammonia is very toxic. It must be removed pretty quickly. Urea and uric acid are less toxic, but they have varying solubilities, and we have to think about what's going to be energetically expensive versus energetically inexpensive. So use this module within the context of osmoregulation to think about what are the needs of the organism, how does that relate to the type of nitrogenous waste that they produce, how could changes in habitat have an impact on their ability or create an inability to effectively generate these wastes, and what might the ramifications of that be.